12th Bio-Zoology. Chapter 5 Molecular Genetics. In this part, we are going to discuss about the steps in DNA fingerprinting. Already we discussed about DNA fingerprinting in the previous video. In this part, we are going to discuss about steps in DNA fingerprinting and its applications. The first step in DNA fingerprinting technique is extraction of DNA. DNA is collected from blood sample, hair, bones, and from different other genetic resources. From the sample, the DNA is separated. The process of DNA fingerprinting starts with obtaining a sample of DNA from blood, semen, vaginal fluids, hair, root, teeth, bones, etc. The DNA is cut into fragments at particular places by using restriction enzymes. Restriction enzymes act as molecular scissors. These enzymes cut the DNA at particular places. DNA is treated with restriction enzymes which cut the DNA into smaller fragments at specific sites. The smaller fragments have to be amplified. Sometimes the sample DNA will be very small, so the DNA number is increased by polymerase chain reaction. Already we discussed about PCR or DNA amplification in Human Genome Project. In many situations, there is only a small amount of DNA available for DNA fingerprinting. If needed, many copies of the DNA can be produced by PCR, DNA amplification. A small piece of DNA is available. It is multiplied by using PCR method. So after getting many copies, the fourth step, electrophoretic separation. We have DNA of different fragment length. We have DNA fragments of different length. These fragments are separated from each other by the process electrophoresis. During electrophoresis in an agarose gel electrophoresis, the DNA fragments are separated into bands of different sizes. The electrophoresis process causes the separation of DNA into bands of different sizes. So electrophoresis, here agarose gel medium is there. DNA sample is introduced in this area. DNA is a negatively charged molecule, so it moves towards a positive electrode. After the movement of DNA of different sizes, we will get separated DNA into bands of different sizes. We will get the DNA into bands of different sizes. Now the DNA bands are found on a gel. The DNA, separated DNA is denatured. It involves separation of double-stranded DNA into single-strand DNAs. The DNA on gel is denatured by using alkaline chemicals or heating. Denaturing of DNA is done by heating or alkaline chemicals. The double-stranded DNA gets separated. Then the separated DNA, denatured DNA is blotted by a process Southern blotting. The DNA bands are found on electrophoresis gel. A nylon membrane is placed on the gel. so that the DNA bands are transferred into the nylon membrane. This is a nylon membrane showing the transferred DNA bands. The bands of DNA are moved out of the gel using a nylon membrane. We have to identify the specific DNA needed for fingerprinting. For fingerprinting, the repeated sequences satellite DNA is separated from the main DNA. For the purpose of identification of the needed DNA for fingerprinting process, probes are used. The probes can also be prepared by using either fluorescent substance or radioactive substance. So probes are DNA sequences. We know the sequences. Probes are known DNA sequences. The probes are coated with fluorescent or radioactive isotopes. This is a probe. Its sequence is known to us. It is coated with radioactive isotopes or fluorescent substance. And these probes are mixed with DNA fragments which have already been amplified and separated by electrophoresis and denatured by using chemicals or heating. The probe with known DNA sequence is mixed with the DNA fragments. Here ATCCGA, the probe DNA pairs with the complementary DNA sequence of the probe DNA pairs with complementary DNA sequence and form a hybrid DNA. We know the sequence of probe so that the complementary sequence can be easily detected. These probes do not attach because these probes do not have complementary bases. So these extra probes are washed off. After probe hybridizes, the excess probe are washed off. A photographic film is placed on the membrane containing DNA hybrids. The nylon membrane contains DNA hybrids. What is DNA hybrid? A DNA probe of known sequence and DNA fragment which is used in DNA fingerprinting. A photographic film is placed on the membrane containing DNA hybrids. When the photographic film is processed, we get the banding patterns. After processing of the photographic film, we get the banding patterns of DNA, thick and thin bands. The thick and thin dark bands form a pattern of bars which constitutes a genetic fingerprint. The application of DNA fingerprinting, the first application is criminal identification and forensics. Whether the criminal is A or B or C, that can be identified. Already we discussed in detail in the previous part. And medical cases, paternity disputes, dispute related to father, maternity disputes, when child or a newborn child are exchanged in hospitals, who is the mother, the problem will arise. To solve the disputed maternity, the DNA fingerprinting is done. Pedigree analysis, to understand the pattern of inheritance of genetic diseases through generations, the DNA fingerprinting of different generation people are taken and pedigree analysis can be done. This is to understand the inheritance pattern of genetic diseases. Wildlife management, illegal trade of protected species is monitored. When people involved in illegal trade of the flesh of protected species, after the DNA fingerprinting of the flesh, the animal can be detected, but if it's a flesh or protected species, that can be monitored and stopped. In Japan, whale meat had been illegally imported with fish products that was detected by scientists. The DNA fingerprint of tissues obtained from dead endangered organisms are conserved. DNA fingerprinting of tissues of dead and endangered organisms is maintained as a record. This is to conserve wildlife and protection of endangered species. Anthropology. Human fossils can be compared easily with DNA fingerprinting technique. The evolutionary history can be reconstructed clearly by using this technique, and this technique will help us to answer questions concerning human origin and human migration, etc. So this technique, DNA fingerprinting, is used in various fields.